my biology classes. This is the first video in the series of genetics. Before this, we had completed 3 series, that is human physio, cell bio and reproduction. Now in this video, our topic is principle of inheritance and variation, and this video is from CBSE class 12th, part 1st of chapter 5th. So at first, in this video, we will start that why the members of the same species are alike. And for this, here, the first thing we will have to know that heredity. So, the heredity is the transfer of information from the individuals of one generation to those of a future generation, commonly through direct genetic descendants called heredity. Genetics is the study of inheritance and variation. So, the genetic is the scientific study of mechanism of inheritance by which the character pass from parents to offspring and the causes of hereditary variations in living organism related by descendant. The term genetics was coined by William Bateson in 1906. Now, the earlier pre-Mendelian theories of heredity, and there are many theories were put forward before Mendel to explain in what physical form the traits passed from the parents to the offspring, and these are called theories of blending inheritance, in which the first one is vapor theory. According to this, Pythagoras proposed that each organ of an animal body emitted some kind of vapor, and that a new individual was formed by the combination of the vapor from different organs. Second is fluid theory. Aristotle held that man's semen was highly purified blood. Woman's menstrual fluid was the female semen, which was not as pure as male semen. So, the two fluids combined during intercourse and the female semen provided inert substance for the formation of the embryo, and the male semen gave form and vitality to the embryo. Next is pre-formation theories. Antony von Leeuwenhoek was the first to observe human sperms. He was then wriggling in the semen and called them animalcules. He held that each sperm was a potential human being and could develop into a new individual when introduced into the woman's womb, where it could get nourishment. After this, another Dutch scientist, that is Swarm and his co-workers, performed miniature animalcule were present in the gamete, and that the development was simply an unfolding and growth of the miniature. Hence, the young ones were after the parents, and the miniature form of the animal present in the gametes was called homunculus. In this picture, you can see the structure of the homunculus. Some workers thought that it was present in spermatozoa and others thought that it was present in eggs. So, these are respectively called spermist and ovist, and the term performationist covers the both. After this, particulate theory. Maupertuis proposed that each animal produced minute particles for the reproduction, and a new individual is formed by the union of the particles of the two parents. The last is theory of pangenesis. Here, the Charles Darwin in 1868 forwarded the theory of pangenesis for the inheritance of characters. He assumed that a tiny representative particles called pangene or gemule by him formed in each part of the body and migrate via blood to the reproductive cells, that is gametes, and hence to the offspring to guide the formation of the respective part. Thus, the young one has a blend, that is mixture of the pangenes, hence of the characters of both the parents. After this, now the Mendelism. And here, the Gregor John Mendel was born in peasant family of the Moravia. Mendelian inheritance, also called Mendelism, the principle of heredity, formulated by Austrian-born botanist, teacher and Augustinian prelate Mendel in 1865. He went to the University of Vienna, where he studied natural history and mathematics for 2 years. He became interested in the process of hybridization. His choice of subjects and his aptitude for hybridization had a significant influence on his later work on inheritance on pea plant. Mendel is known as father of genetics because of his laws from the basis of the science of genetics. After this, here the Mendel's experimental plant. And here you can see the Mendel worked on garden pea, that is edible pea plant, which is known as Pisum sativum. He selected these plants because of the following special advantages. First one, that is the garden peas were found to differ in certain definite and easily detectable traits. So, the Mendel selected 7 pairs of characters which have contrasting traits, and the traits which appear in two opposing conditions, one dominant and other recessive, are called contrasting traits. So in this picture you can see there are 7 characters, that is seed shape, seed color, pod shape, pod color, flower location and the plant size, in which the round, yellow, inflated, green, purple, axial and tall all are the dominant characters, while the rest wrinkled, green, constricted, yellow, white, terminal and the dwarf are the recessive characters. The traits of each kind of the pea plant were preserved in generation after generation because the plants had bisexual flowers and normally resorted to self-pollination. Artificial cross-pollination could be easily achieved by removing the stamens before the pollen grains matured and dusting the pistil of this flower with the pollen from a desired plant. So in this picture, the parent, here the removal of the anthers, this is known as emasculation. After that, the transfer of pollen, that is dusting. From this, the new, after the artificial cross-pollination, new plant will be formed. Pea plants being annual had a short life cycle. Pea plants produced many seeds in one generation. It could be raised, maintained and handled conveniently. Mendel worked on only pure or true breeding traits. So, the pea plants having each of the 7 characters he selected were readily available. Now, the Mendel's experimental techniques. And here you can see Mendel's conducted breeding experiments in 3 steps, in which the first one is selection of pure parent plants. Mendel first confirmed that the traits of 7 characters noted by him were true breeding, that is, they were passed on from parents to offsprings. Here you can see the 7 pairs of contrasting characters of the Pisum sativum chosen by Mendel for his experiment. Here you can see the dominant traits and the recessive traits both. The second is hybridization of pure plants. Crossing or mating of 2 varieties of the plants or animals is known as hybridization. So, the pure breeding plants were used by Mendel in his hybridization experiment, and in these experiments he crossed pure plants having contrasting traits of the character. And after this, the flowers will be pollinated by the hand were kept enclosed in paper bag from the very beginning to avoid entry of unwanted pollen. So, the seeds formed by crossing the plants were collected and the plant raised from them next year. They produced hybrid plants that constitute first generation. The first generation of the Mendel cross was named the first filial generation, which was indicated by F1 and given by Betson and Saunders in 1902. And the third step is self-pollination in hybrids. The hybrid plants of F1 generation were subjected to self-pollination. For this, their flowers were enclosed in paper bags to keep out foreign pollen, and the seeds produced were collected and sown. They produced F2 generation called second filial generation. Self-pollination in the F2 gave rise to the F3 generation called third filial generation. After this, the Mendel's monohybrid cross, that is inheritance of one gene. A monohybrid cross is the hybrid of 2 individuals with homozygous genotypes which result in the opposite phenotype for a certain genetic trait. The cross between the 2 monohybrid traits, that is capital T and small t is called a monohybrid cross. It is responsible for the inheritance of one gene. It can be easily shown through a Punnett square. It is used by geneticists to observe how homozygous offsprings express heterozygous genotypes inherited from their parents. Now, how to carry out a monohybrid cross? So, for this, it indicates the alleles using characters, that is, recessive alleles can be indicated by lowercase letters, while the dominant alleles can be indicated by uppercase letters. Both the phenotype and genotype of the parents or the parental generation that are being crossed. The genotype of the gametes from the parental generation as a result of meiotic division, the gametes will be haploid. Tabulate a Punnett square to check out the probable combinations of the gametes. Any combination is possible in the process of fertilization is random. The phenotype and the genotype ratio of the prospective offspring can be written. The outcome hence obtained is known as the F1 generation. The F2 and F3 etc. generations form the subsequent generations. Now, the example of monohybrid cross. And here, you can see here, for the monohybrid cross, Mendel began with a pair of pea plant with two contrasting traits, that is one tall and other dwarf. The cross-pollination of tall and dwarf plant resulted in tall plants. All are hybrid and called F1 generation. He conducted an experiment with all 7 contrasting pairs and he observed that the entire F1 progeny showed one pattern in their behavior and the another pattern character was completely absent. He continued his experiment with self-pollination of F1 progeny plants and surprisingly he observed that 1 out of 4 plants were dwarf while the other 3 were tall. So, the tall and the dwarf plants were in the ratio of 3:1 and called F2 generation. So, the Mendel observed that traits which were absent in F1 generation had reappeared in the F2 generation. And here, the inheritance of single trait is called the monohybrid inheritance. Now, the reciprocal crosses. A reciprocal cross involves the same traits but carried by sexes opposite to those in the original or first cross. Like if in the original cross, pollen from the flower of a tall pea plant were transferred to the stigma of the flower of a dwarf plant. In a reciprocal cross, pollen from the flower of a dwarf plant are deposited on the stigmas of the flower of a tall plant, just opposite of the original crosses. Now, here you can see all the characters in the F1 generation. Here, all dominant characters appear in F1 generation, but in F2 generation, approx 3:1 ratio of dominant and recessive characters appear. So in this picture, all the 7 characters, their contrasting traits and the F1 results, then the F2 results and F2 ratio, approximately 3:1 ratio of all these characters. Now, the findings or results of the monohybrid cross. So here, according to the results of the monohybrid cross, F1 plants produced by a cross between 2 plants pure for the contrasting traits of a character are similar and express only one contrasting trait. It doesn't matter which parent provide male gamete and which provide female gamete, the result remains the same. Both the contrasting traits appear in the F2 plants. The contrasting traits of a character expressed in F1 plants appear in 3/4 of the F2 plants. One of the 2 contrasting traits of a character remains unexpressed in F1 generation. Now, the conclusions drawn from the monohybrid cross. So here, the gametes bring from the parents something which makes a character appear in the next generation, and this something was called by him a factor. The inheritance characters are thus determined by separate unit factor. There are a pair of unit factors for each character, one inherited from each parent. For instance, height in pea plant is controlled by 2 factors, since some plants are tall and some are dwarf. The Danish botanist Johnson used the term genes for the Mendel's unit factor in 1909. Third, that is, certain factors do not express their characters in the individual, but some factors do not change and may express in the later generation. Now, the law of segregation. Here, the factors of each character segregate during gamete formation, so that each gamete receives only one factor for each character and is always pure, and thus two factors of a trait occur with equal frequency in the male and female gametes. So this is the Mendel's law of segregation. Now, the traits do not blend according to the monohybrid crosses. So, the Mendels had so noted that none of the plants in F1 generation was intermediate in size between the two parents. From this, he assumed that blending of characters do not occur. The two factors of a character are similar in individuals which breed true and are dissimilar in the individuals that do not breed true. Mendel found that expressed ratio of 3:1 is actually 1:2:1. Here, this 3:1 ratio is called Mendel's monohybrid phenotypic ratio and 1:2:1 ratio is called Mendel's monohybrid genotypic ratio. Now, the Mendel's principle of heredity on the basis of genes. So there are two fundamental principles, that is law of segregation and law of independent assortment, framed by Mendel, are universally true and they are called first and second law of Mendel. Except these, there are two other principles, that is law of unit characters and the law of dominance, though of prime importance in heredity but are not universally applicable. So here these two are, first one, that is the principle of paired factors. It states that a hereditary character is controlled in a diploid organism by at least two alleles that occupy the two corresponding loci in the homologous chromosomes. So in this picture, you can see the principle of paired factor. An individual receives one allele for a trait from each parent and passes one of the two alleles for a trait at random to each offspring. The two alleles for a trait are similar in a homozygous individual, like as an example, here the capital P is a pure purple flower and are dissimilar in a heterozygous individual, that is capital P small p, which is a hybrid purple flower. Now, the principle of dominance. It states that one allele of a pair of alleles which control a character usually express itself and prevent the expression of others. The allele which is expressed in a hybrid is called a dominant allele and the unexpressed one is termed recessive allele. So here in this picture you can see the Mendel's law of dominance. And here, the cross a homozygous tall P plant having the genotype T with a homozygous dwarf P plant having the genotype t. In F1 generation consists only of tall plants having genotype T. Since they have an allele for dwarfness also, they are all hybrids. So, the expressed allele T for the tallness is dominant over the unexpressed allele t for dwarfness. The fact that the allele for dwarfness is present in the F1 plants can be verified by inbreeding them when the F2 progeny will consist of both tall and dwarf plants in the ratio of 3:1. So, this is the principle of dominance. Now, the Mendel's first law, that is the law of segregation. And here it states that the two alleles controlling each character maintain identity in the organism but during the formation of gametes or spores by meiosis move apart due to separation of the homologous chromosomes which bear them, so that each gamete or spores receives only one allele of each character on random basis. Since the gametes or spores possess one allele of each character, they are always pure. So this is also known as purity of gametes or spores. Now, in this picture you can see this law that the pair of alleles in P generation segregates from each other during meiosis, that is cell division, and here the only one allele will be present in each gamete. In a monohybrid cross, both the alleles are expressed in the F2 generation without any blending, and thus here the law of segregation is based on the fact that each gamete contains only one allele. So this is known as the law of segregation. Now the explanation of law of segregation, and here you can see the cross between the guinea pigs. Here in this picture you can see in original cross or the reciprocal cross, white and black guinea pig shows law of paired factor, that is alleles are in pair. Then according to law of segregation gamete forms and so fertilization to form F1 generation where all are hybrid black. Then the second fertilization occur and it shows the law of dominance where the 3:1 ratio found. So 3 are black in which 1 is homozygous black and the 2 are heterozygous black, while the other one is, that is 4th is homozygous white. So this is explanation of law of segregation. So the allele behavior in F2 generation of a monohybrid cross, here it shows the allele distribution into gametes and they are coming together in the offspring. A gamete recessive only one of the two factors and the genes of a trait. Therefore, the 50% of male and the female gametes formed in F1 get the factor or gene for the black hair and the other 50% get the factor or gene for the white hair. Because of this, the ratio is, in F2 ratio is 1 pure black, 2 hybrid black and 1 pure white. In F2 generation, the phenotype ratio and the genotype ratio are different. The phenotypic ratio is 3:1 whereas the genotypic ratio is 1:2:1. Now, the Punnett square. If the genotypes of the parents are known, the genotypes of their result offsprings can be easily predicted with the help of a chart called Punnett square. It is named after an English geneticist, that is Reginald Crundall Punnett. Here, its advantage lies in the minimizing the errors which can creep in when listing all possible combination of gametes. Each box of a square represents an individual offspring, and the alleles of all the male gametes are entered in the left vertical square, and then the alleles of all the female gametes are entered in the upper horizontal square. So, now the remaining boxes are filled in by combining the alleles of males and females gametes, working horizontally or vertically in each row. So the allele combination in the boxes show the possible genotypes of the F2 generation and the ratio of genotype can then be worked out. So this is the Punnett square. Now the Mendel's dihybrid cross or the inheritance of two genes. A breeding experiment dealing with two characters at the same time is called dihybrid cross. In one such experiment, Mendel considered form of the seed and color of the cotyledons. In a dihybrid cross, the parents carry different pair of alleles for each trait. One parent carries homologous dominant allele while the other one carries homozygous recessive alleles. So the offspring produced after the crosses in the F1 generation are all heterozygous for specific traits. This is the dihybrid cross. Here the two characters. Now, Mendel took a pair of contrasting traits together for crossing and like color and the shape of seeds at a time. He picked the wrinkled and green seed and round and yellow seed and crossed them. He obtained only round and yellow seed in F1 generation. This indicated that round shape and the yellow color of the seed are dominant in nature and the wrinkled and green are recessive in nature. Then the F1 progeny was self-pollinated and this resulted in 4 different combinations of seed in the F2 generation. They were wrinkled yellow, wrinkled green, round green in the phenotypic ratio of 9:3:3:1. Here, Y consider for the yellow seed and the small y for the green seed color, while the capital R for the round shape seed and the small r for the wrinkled seed shape. Thus, the parental genotype will be capital Y, capital Y, capital R, and small y, small y, small r, small r. So, this is an illustration of the dihybrid cross. Now, you can see here all the genotypes and phenotypes formed in the F2 generation. Here, character is seed coat and the cotyledon colors, and the traits is round wrinkled and yellow green. And the alleles are capital R, capital Y and small y. You can see here the characters, traits, alleles, genotypes and the phenotypes. So there are 9 genotypes but only 4 phenotypes are present. Now, Mendel actually got 529 seeds from F2 plants, and these include 301 round and yellow seed, 102 round and green seeds, 96 wrinkled and yellow, and 32 wrinkled and green seeds. He found that plants with yellow and green cotyledons were in the ratio of 12:4 or 3:1, and the plants with round and wrinkled seeds were also in the ratio of 12:4 or 3:1. Now, the finding or results of a dihybrid cross. So, the 4 types of plants. A dihybrid cross produced 4 types of plants in F2 in the ratio of 9 with 2 dominant traits and 3 with 1 dominant and 1 recessive traits, 3 with other dominant and other recessive trait, and the 1 with 2 recessive traits. And the new combinations. 2 new combinations of traits, that is round green and wrinkled yellow, had appeared in a dihybrid cross. Now the conclusion drawn from a dihybrid cross. The inheritance of cotyledon color was independent of the inheritance of the seed shape. This led him to formulate 4th law of heredity. This is the law of independent assortment. Now, the Mendel's second law, that is the law of independent assortment. This law states that the alleles of different characters located in different pairs of homologous chromosomes are independent of one another in their segregation during gamete formation and in coming together into the offspring by fertilization, both processes occurring randomly. According to the explanation, this law can be explained with the help of dihybrid cross, which you can see in the picture. Here the example of a dihybrid cross, that is the Guinea-pigs, which shows the law of independent assortment. So, here the parents which has phenotype pure black and short haired, whose genotype is capital B, capital B, capital L, crossed with pure white and long haired, whose genotype is small b, small b, small l, small l. Then the gametes will be formed by the law of segregation, and one out of each pair of each character come in one gamete, that is capital B, capital L and small b, small l. Then after the fertilization, capital B, capital L, small l form, which shows F1 generation and all are hybrid, black and short haired. Then in F2 generation, they form, because of the dihybrid cause, 16 offsprings, which shows 9 black short haired, 3 black long haired, 3 white short haired and 1 white long haired. So here the offsprings are formed in the ratio of 9:3:3:1. Now, the 9:3:3:1 dihybrid phenotypic ratio is the product of two separate 3:1 monohybrid ratio, one for each allele pair. The dihybrid cross dealing with the two characters simultaneously yields the same result as does a monohybrid cross, namely. In F1 generation, raised for the two pure plants, all offsprings are alike and resemble the parent with dominant character superficially. And in F2 generation, the dominant and recessive characters appear in the ratio of 3:1. This proves that the alleles of different characters do not interfere with one another's behavior but are sorted out independently of one another. Now the limitations of the dihybrid or independent assortment cross. It holds only if the different pairs of alleles lie in the different chromosome pairs, since it is chromosomes and not individual alleles which are segregated during reduction division. Now, here all the three principles of the Mendelian inheritance, like the law of segregation in which the two alleles for each genes are placed in different gametes. Second is law of independent assortment. The inheritance of one gene doesn't affect the inheritance of any other gene. And the third is law of dominance. When the two different alleles are present, only one is dominant and will be expressed. Now, the Mendel's trihybrid cross. Mendel conducted trihybrid crosses that involves three characters simultaneously. Now the summary of Mendel's hypothesis. Each trait of an organism is controlled by a pair of alleles. In an organism having two unlike alleles for a given trait, one may express and the other may remain unexpressed. The two alleles of a trait separate during gametogenesis so that each gamete receives only one allele of each trait. During gamete formation, the allele pair of the different traits segregate independently of each other. Each allele is transmitted from generation to generation as a discrete, unchanging unit. And each organism inherits one allele for each trait from each parents. So this is the summary of Mendel's hypothesis. Now, the reasons for the Mendel's success. So, the many plants and animals hybridists had conducted experiments before Mendel's, but they failed to arrive at clear-cut rules of heredity due mainly to not keeping precise records of the offsprings produced by various crosses. So, the Mendel succeeded in formulating the basic principles of heredity due to following reasons, in which the first one is Mendel's experimental plant garden pea is ideal for controlled breeding and it can be easily cross-pollinated. Mendel selected true breeding pea plants for his experiments. He studied at a time the inheritance of a single trait with two contrasting conditions instead of attempting to study the complete inheritance of an organism. He kept an accurate record of his breeding experiments and noted down the number of each type of offspring produced in each cross. He pulled the data of many similar crosses and analyzed the result by using statistical methods and the law of probability. He was lucky in selecting 7 particular characters of this plant. Each character Mendel studies is determined by one gene, for which there are two alleles, one completely dominant to the other. And the last is the traits did not show interactions, incomplete dominance, linkage, etc. So, the principle of heredity laid down by the Mendels are not found to be applicable in it can completed cross of inheritance and also with the slight modifications. And the study of Mendel's law of heredity is called Mendelism. Now, the explanations of some important terms that are used in genetics, in which the first one is gene. It is the unit of inheritance which is carried from the parent by a gamete in a chromosome and controls the expression of a character in the young one in cooperation with its allele, other genes and environment. Second is allelomorphs, that is alleles, a pair of genes that control the two alternative expressions of the same character and have the same loci in the homologous chromosomes were originally called the allelomorphs or alleles. Next is gene locus, a specific region of the chromosome representing a single gene or allele is known as gene locus. And here wild and mutant alleles. An original allele dominant is expression and the widespread in the population is called wild allele. An allele formed by a mutation in the wild allele recessive is expression and less common in the population is termed mutant allele. Now the difference in dominant and recessive allele. So, the dominant allele can express itself in the presence as well as absence of its recessive allele, while the recessive alleles can express in the absence but not in the presence of its dominant allele. Dominant allele codes for a complete functional polypeptide that expresses it fully, while the recessive allele codes for incomplete non-functional polypeptide which fails to express it in heterozygous condition. Now the gene pool. All the genotypes of all the individuals in an inbreeding population make up the gene pool. Now next is genome, and here a complete set of genes contained in the haploid dose of chromosome and inherited as a unit from one parent is called genome. Next is pure lines, a strain of genetically pure true breeding organism resulting from continued selfing or inbreeding is called pure line. Next is F1 and F2 generation, that means first and second filial generation. F1 generation of hybrid offspring produced in a genetic cross between the two genetically different but pure parents, like crossing between the T and the small t parent P plant form T small t in the F1 generation. And the F2 generation of young ones produced in a genetic cross between the two F1 organisms. And these terms F1 and F2 are introduced by Betson and Saunders in 1905. Now, here the difference between the homozygous individual and the heterozygous individual. Homozygous individuals have similar alleles for the specific trait, while in the heterozygous individual have dissimilar allele for a specific trait. Homozygous individuals possess dominant or recessive alleles, never both, while the heterozygous individual possesses both dominant and recessive allele. Homozygous individuals produce only one kind of gametes, while the heterozygous individuals produce two kinds of gametes. Homozygous individuals breed true for a specific trait, while the heterozygous individuals do not breed true. Homozygous individuals do not have additional vigor, while the heterozygous individuals have additional vigor called hybrid vigor or heterosis. So, the homozygous are pure individuals and the heterozygous are hybrid individuals. The homozygous and heterozygous individuals are called homozygotes and heterozygotes respectively, and these terms were introduced by Betson and Saunders in 1902. Now, the phenotype and genotype. So what is phenotype and what is genotype? Phenotype is the expression of the trait, while the genotype is the gene equipment that determines a trait. Phenotype can be noted by the direct observation of an individual, while the genotype can be ascertained from the ancestry or progeny of an individual. Phenotypic individual having similar phenotype may or may not have similar genotype, and the genotype individuals having similar genotype develop similar phenotype in the same environment. Phenotype may change under the influence of environment, while the genotype doesn't change under the influence of environment. Phenotype determined by genotype, while the genotype not influenced by phenotype. So, the T, small t, and T small t are the genotypes for the size of stem in the pure plant, which are tall, dwarf, and hybrid. And here, the individuals of different genotypes may also have the same phenotype, like the T and T small t give rise to tall pea plants. So this is the end of this lecture, in which you understand about the Mendel's three laws, that is law of dominance, law of segregation and law of independent assortment, and monohybrid cross and the dihybrid cross, and the basic terminology related to the heredity. Now the next video will be on the chromosomal theory of inheritance. So if you like this video, so like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.